Check, 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 check. Can you hear me now? All right, after our Verizon interlude, today we're going to take a look at the biggest organelle that's there in cells. And what should have been obvious from that opening DIC sequence of living cells in a dish moving around is not only are we trying to emphasize that these cells are alive, and if you view them in time lapse, you'll at least psychophysically look at them as if, oh, I can recognize that as life. It's going somewhere, it's doing something. And in the middle of each of those fried egg-like profiles was a bump. And the bump was the nucleus. And it's what actually differentiates between prokaryote, those before the nut, and eukaryote cells. All of us are composed mainly, in our body, somatic cells, by eukaryote cells. And they're mainly diploid cells. And we'll get to that at the end of the lecture. But in the initial days of microscopy, then, looking at something like this, and here we have what's the view of an epithelia here, a couple ducts whose name aren't important until at least tomorrow. And in these, we have, let me see if I can make it so that this doesn't turn itself off. In these, we can see there's an open lumen, and in the usual stain, hematoxylin and eosin, which I promised you was going to be there 95% of the time, we can make out in the center of each of these columnar cells a really bright pinkish spot. And some of you up close may be able to see that it's heterogeneous, it's not uniform color. And in these this is basically the nucleus, right? And you realize that much of the information that then programs the outcome, the phenotype, what you see here before you, is dictated by what's going on in this nucleus and the genetic information that's within it. One important thing that you shouldn't forget as we go along in this is that during moments of cell division, and we're going to be looking at two types at the end of the lecture, mitosis and meiosis, there's going to be a replication and a sorting of this genetic information. Fine. There's also going to be a replication of all the other organelles that are there, and that part of the environment around this nucleus gets propagated on its own. All right? So there are things that are genetic phenomena, and there are things that are epigenetic phenomena that come from modifying what parts of the genome get expressed and why. All right? So Lamarck wasn't completely wrong. He just was wrong about that giraffe being able to stretch its neck. There are things that will happen in your generation that will affect the F2 generation down the line, okay? So, what we expose ourselves to in the environment matter. But today we're going to take a look at that nucleus, and we can take a look at these pink spots then at the EM level, and we see something entirely different that I used in that first lecture. And it may be sort of difficult to make out, but right here is this huge profile of the nucleus. And you can see, compared to things like the ribosomes and a mitochondria here and the Golgi stack, it really is enormous in this cell, all right? Now, this is one particular cell type, and there are others as well. And one thing you may be able to remember from, I hope, the last couple of days is that this structure that you see multiple reproductions of is what? Rough ER. Okay, so this cell's deeply involved in protein production and probably excretion, et cetera, all right? That's what this cell is set up for. But you can see other things within this. You see, obviously, the nucleus, the subject of today's lecture, and you also see, basically, big fat droplets. And right along in here, you may be able to make out that these structures, cut at various angles, have what appear to be partitions in it. And these are mitochondria as they really appear, not those sort of schematic things that we always draw you looking like a bacteria. Those, In fact, mitochondria in each of the different major tissue types are different. And you know from an electron micrograph of the formation of the crista and the rest of these things in it, you could tell which tissue you're looking at. You also find that they're bifurcated, or they may fuse together into long things. They have a life cycle of their own. They move around. They go toward areas of high calcium in cells. They're really active, all right? So in your mind's eye, take an electron micrograph like this and think of things moving around, doing stuff. Just like you saw in that differential interference contrast picture, we've got stuff here, and you just have to have the imagination to embrace it. Now today, we're going to look at this particular organelle in detail, and you might be able to make out that first, it does have a certain heterogeneity that's there, and second, there seems to be a boundary around it that I'm trying to follow here, not successfully on my trackpad. So if we take a look at that in more detail in a different cell, a higher magnification, and you can tell it's a different cell, it has a different formation in terms of this ER that's there and the rest. And one of the important things that we have to do is sort of name the parts and try to figure out what's the function that goes in along with these structural elements that are there. So around this nucleus, which is sitting there halfway on your screen then, you can see the lighter areas of faint stippling and then these darker areas. This lighter area is euchromatin, EU chromatin, and it is areas where things seem to be spread out, and you might imagine that functionally it's places where we're actually taking that genetic information and we're going to translate it out for producing something. Areas which are denser, like shown along here, along basically this sort of partition between the nucleus and the cytoplasm, are more condensed. Okay, and that's heterochromatin, and what you see is what you get. Depending on the state of the cell, what it's doing, it'll have more or less of these two components of heterochromatin where it's condensed there, or euchromatin where it's dispersed. What you also might be able to make out here down at the bottom of the image, and you can see some granularity in there, is, every organelle should have an organelle, right? So this is the nucleolus by extension down. And we're going to look at it in particular because what it does is the translation of our ribosomal RNA. Okay, so we're going to be looking at it. Hello? I've got the arrow as big as it goes on my screen, and now that it isn't getting big here, I'm going to find out from Lee why it doesn't scale, because it's about a quarter of my screen. So I feel sort of like Dumbo down here, and then I look up there, and it's not there. My alternative would be to use a pointer, and then you'll find that on the Echo being just a dash, a dot that moves around the screen. You won't be able to see it. So for today, I'll try moving the arrow very slowly. And like all good pouncing cats, you should be able to find it. Yes? Can you point to the nucleolus again? I'm sorry? Can you point to the nucleolus again? Yes. The question was, could I point to the nucleolus again? Now, should I? Yes, I can. I will. So the nucleolus is this, we have a condensation that's not around the outside of the nucleolar wall, right? And you can make out down in this part right here, there's some heterogeneity. And we're going to see that on its own in a big expanded version in just a bit. Okay, now the other thing that I want to point out is not only do we have occupancy here of the nucleus, but if I move very slowly up here, you notice it's hard to distinguish between the wall of this ER and the wall of the nucleus. In fact, it looks like it's folded over on itself, except at areas like here, or if I go over here, there are breaks in it. Okay, so we have two functional problems that we have to solve. One is, what is the wall of a nucleus? And second is, what is that hole and what can go through it by implication? And does this going through involve something that's passive diffusion, or is it something that's active transport? All right? Because as you know from the plasma lemma, the cell wall itself, we have a lipid bilayer that's surrounding that. And you may or may not remember that this ER is also a lipid bilayer. So it's suggesting from this image, at least, that this folding over around near these pores is giving us something that's a folded two-layer sheet of lipid bilayer, right? So there's 4 layers of lipid creating this nuclear membrane. And the question is, well, it's associated, it's near there, but indeed, what is this pore-like, and why does it appear to have a little line running across it? What's there? Well, let's look at the schematic that's in your book. And by your book, I do mean version 6 of this. And version 6 really matters. I know there's some really kind M3s and M4s that are trying to sell you version 5. How different can it be? It can be really different. In other words, we rewrote the manual to reference the pages that are in here. And if any of you think that it's a good idea to spend your time trying to find the counter pages on it, good luck, because about 25% of the pages in here are new and different. So you're not going to find them. Okay? So whatever you do, since the lab manual and the course rely on it, get version 6 of the book. This is a figure from that, and we can make out here, this is showing you a rough ER, and these are supposed to be ribosomes sitting in there, and then the lumen of this, which contains a whole bunch of good things like the proteins you're producing and calcium environments, et cetera, is surrounded by this lipid bilayer. And here's where I'm showing you that the lipid bilayer folds back upon itself to produce what is that major wall surrounding the nucleus. It then has some fenestrations in it, shown here in green, here and there. And those are the nuclear pore complex. And we'll look at that in more detail, both at the light and the EM level. Now, underneath this surrounding of lipids, right, is actually a nuclear lamina, which is shown here as sort of a mesh network, and that nuclear lamina is composed of laminins. And the laminins are what effectively give it structure, and you'll find that chromosomes, the telomeres of chromosomes, sit there and they attach themselves at various points in the cell cycle to this network that's on the inside. The scanning electron micrograph, the SEM of that lamina, is shown spread out here on the right-hand side. Okay, so take a look over here for that. And the natural functional question comes up. Remember, we're going to talk about cell division at the end. And you realize that if I have basically 2 kinetic cores that are sitting there and being pulled apart by some organizing feature and microtubules and their motors that Dr. Brady referred to, that to be able to pull apart those things, I better make the nuclear membrane disappear. Right? It's got to go out of the equation for a moment. I have to become a prokaryote again, so that I can take that genomic material and take it to either side and then collapse the cells into 2 daughter cells and basically reform that membrane. So the question is, where does it come from? The evidence is that, as you might expect, that these ER on its own can reconstruct at least the lipid component of this nuclear envelope. Okay? And that is demonstrated then if we look at another video, and in this case one in which we've taken basically microtubule lipids, and they're lying out here in a dish, and asked what will they do spontaneously. And what they do spontaneously is rather incredible, because they're going to actually sit there and start to fuse. In an area that has laminins on it, they will not remain as whatever they would be out in the cytoplasm, being ER. As you can see, where we get these junctions of larger and larger aggregations, they are forming what is going to be that double bilayer, all right? And they basically give yourself an example of something that you might plausibly believe is going to be the nuclear envelope, okay? So, you know, some of these things during the division spontaneously organize themselves. So, let's go back and talk about those holes. And boy, do I wish my arrow were a little bit smaller. Let's talk about then up here at the top is a schematic, and here is a view looking from the nucleus out at these pores. And these particular scanning electron micrographs are made by, the initial ones were made by one of these 1,000,000 electron volt microscopes, taking the water away, so it's basically been frozen in place, dehydrated, and then viewed destructively, but at least able to capture these images. So what it looks more or less like is, you know, sort of some strange lobster basket or something on the inside that's there. And if we were to take a look at the outside, it has sort of tendrils that go up from it. Around the perimeter, then, are sort of like staves of a barrel on this. And they have components which, remember, this is a double lipid membrane, right? They have components that are sitting into that membrane, holding them in place. And in this case, then, the question is, this looks like a pretty big hole, and it is. This is a 50 nanometer scale bar that's down here at the bottom of the picture. And so this opening looks like it's about 50 nanometers. That's pretty big. You know, if you think of lipid membranes between 5 and 8 nanometers, this is a pretty large thing. If we take a look at that then from either side, this is looking at it from the cytosol side, another scanning electron micrograph looking down the barrel of it, and it's from this plane, and versus down at the bottom, which is the view that you just had, you can see that this is the entire interior of this nuclear pore, trying to give you the schematic arrangement to it. Now, I mentioned these were from high voltage electron micrographs. You can actually then tag fluorescently, and using higher resolution light microscopy than what we've talked about, you can actually see these elements of the nuclear pore, the lamina itself underneath, and the DNA. You know, we can take things like DAPI, which intercalates itself inside the minor groove of a DNA double helix and sits there. We can use those three different things colored to see what's on the next slide. Over here is a light micrograph then. Up at the right, at the top, you see a bunch of little cranberries that look like they're sitting there on top of this. Those are the nuclear pores, right, giving you the localization of what's there. What's labeled in green then is lamin B, which is part of this lamination, this network that's just underneath the lipid. And the deep blue that you see here is the DAPI. That's staining then the DNA, the chromatin that's inside it. All right, so it's possible in both these to take a look. This is from a paper in Science in 2008. I mean, it's a rather recent advance. Yes?
Can you go back to the other slide and explain which one is on the outside of the nuclear?
Sure. So, oops. I have one of those magic trackpads that I've set up to do too many things. Here we have this view from the outside, and the arrow is to indicate that it's on the cytosolic side, because this is the cytosolic and this is the nuclear. And this view going across here is on the nuclear side, you know, from the nucleus looking out. Okay. So let's ask ourselves what needs to get in and out on a eukaryote cell from the nucleus. Do we need the DNA to go out there and recombine with things? You know, we are not a prokaryote. We do not have the genome out there waiting to be recombined with whatever Monsanto wants to recombine it with and insert it in something else, all right? That's not us. But we do have a major concern about having, when appropriate, various proteins that are produced. In fact, we don't have any protein synthetic machinery that are inside that nucleus, so we better have a way of getting the information out to the cytoplasm and getting the resulting proteins imported back into the nucleus. So we have two major problems. One of import, what gets to go in, and export, what gets to go out of the nucleus. And that's shown just, you know, as a dendrogram, an outline here in this slide. And we have the major element responsible for protein production, obviously, is the ribosome. And there are two major flavors of them, one that are just free in the cytosol and the other that are attached to that ER, ergo the rough ER. And so these out here in the cytoplasm are responsible for producing the proteins that are going to be going into your mitochondria, into peroxisomes, and bingo, right here, what's going back into the nucleus, what's needed for it. Alternatively, then, those things where the ribosomes that associate themselves with the endoplasmic reticulum are going to be involved in things that eventually work out into the Golgi. Okay, and from being out in the Golgi, they may go off into lysosomes, they go into secretory granules, and obviously it's for the proteins that we want to either, secretory vesicles means we want to release them, or into the plasma membrane, we want them to sit there on the surface and do something for us, whether it's signal transduction or whatever. Okay, so these are the major pathways, and I mentioned that we were interested in the nucleus, so it's going to be this pathway that we want to know about, and that brings up then what sorts of things get imported into the nucleus. Well, the lamins, those things that made the nuclear lamina, they get imported, all right? They weren't produced in there. Histones, which will come up halfway along in the lecture, which is something that we're going to basically wrap our DNA around, they get produced there. The polymerases, and all sorts of things for repairing these things, specifically the RNA polymerases, the ribosomal RNase polymerase, for example, is going to be used to construct the components of those ribosomes, all right? So all that has to be imported in there, and obviously to get signaling and response of what happens at the cell surface to expression in the genome, we better have a bunch of transcription factors that can make it in. Similarly, the T, M, and RNAs must be able to go out, and like I said, the ribosomal particles, we construct what we need, we break it in half and put it out as two particles and construct the ribosome itself back out there in the cytoplasm. So it's sort of complicated, but this gives you a flavor of the sorts of things that are going in the two different directions through this pore. Now, what does that mean in terms of, the cartoon version of reality? Cartoon version of reality here then gives you the idea of this pore. And basically, anything that's smaller than about 40 kilodaltons can go in by passive or out by passive diffusion through these pores, all right? It's the stuff that is larger than 40 kilodaltons, shown over here on the most right-hand side, that are those that need the transport elements. So what does that look like, and how do you argue the case? I'm standing up here telling you lies without fear of retribution, but there actually is evidence that this is true, all right? I'm not just making this up out of whole cloth. And if we take something that is like a normal importing signal that's mutated, all right? So a T antigen that is shown up here, sequence is shown up here, and these little round circles in each of these cases are the nucleus in the cell whose body you can't see, okay? We have fluorescence of just these sort of spheroid things, which are the nuclei. But if we mutate just a single amino acid, what you can see is that that same signal is now sitting out here in the cytoplasm, and it appears that the nucleus is dark. Now, for those of you up close enough to see that there's some haze, remember there's some cytoplasm that's over the nucleus, all right? And this is not a confocal image. We've not taken an optical slice through it. We're just taking a fluorescence picture. The point of this is that this has basically blocked it so that this signal can't make it into the nucleus. All right, now if you don't like arguing at the light level, you can argue at the EM level, and you can use, instead of fluorescent molecules, you can use gold particles, and you can also see here is a nuclear membrane, here's the folding over of that ER doublet, and then here is a pore, and here's a pore. What's being shown in these arrowheads, then, are these very, very small gold particles that are used as your marker. And this is a time sequence from 10, 30, 40, 50 minutes. And the point is you can follow at the electron microscopic level these particles moving from the cytoplasmic side down here into the nuclear side of that pore. Now, it's okay for all of you who can put together a bunch of static images and figure, yes, logically, this must be the case. And it's a very labor-intensive way of doing it. Another way of doing it, and this is looking at a bunch of, in the immune system, a bunch of immune cells and basically a transcription factor that's going to be leading itself into the cell when there is an initial signal at the cell wall. And in this case, the signal at the cell wall elevates cytoplasmic calcium. Now, we can do it either by putting a bug in the dish that these cells will respond to, or we can use a calcium ionophore just to artificially raise the roof on this. Let's go back to it. And if we do that, then you see the sequence in the following slide come to life, I hope. At some point here, we have a GFP fusion protein that is attached to our transcription molecule. We're going to add then this ionophore into the bath, and you'll see the signal moving from the cytoplasm into the nucleus, all right? So that was something that under normal conditions was being held outside of the nucleus and then basically gets concentrated. If we remove the calcium level falls back down. This molecule gets exported, all right? And so we get rid of the transcription factor. It says we don't need more of whatever is being produced in the nucleus. So here's the take-home message. You have a nuclear pore, but you should carry around an image with you, which you'll explore in detail in biochemistry in the coming weeks, is the fact that there are both shown here on the left-hand side of the schematic an import signal cascade that allows big things to bind to a receptor, get transported in through this pore, and an export signal cascade that's here. So it is possible to, at least for these larger molecules, things above 40 kilodaltons, to actually regulate whether or not they can approach the genome. So now we have the functionally significant idea that, okay, maybe we separated off the nucleus because we wanted to actually restrict the conversation that can go on. If we were a prokaryote, whatever happened, whatever flows through the cytoplasm, boom, it can have access to what's going on in your genome. In this case, there's a gatekeeper, all right? And there's a gatekeeper basically that's formed by this pore and the associated elements with it. Now let's switch gears for a second. We've been talking about the nuclear membrane. Yeah?
First, I have a question about the calcium. Do you need calcium with the NLS and the GTPase in order to import things from the cytoplasm into the nucleus?
This was a very specific reaction in this case. In general, no, there's a whole bunch of different signaling cascades. Calcium, as some of you may know, about 50 years ago was the internal messenger, right? It was the first one discovered. And Roger Chen made a living and a Nobel Prize by coming up with reagents that could track it. But then what you find is that it's then integrated in a really complex way with a whole bunch of other intracellular mechanisms and IP3 and other cascades. And they co-release each other and do all the rest. So some of us just stay at the membrane because we can say this is inside, this is outside. And people who want to lose their mind go and devil in the details, look at the interior. Yeah?
The smaller molecules, can they just move freely anywhere on the nuclear envelope, or do they still have to pass through the pore?
They still have to go through the pore. Remember, a lipid membrane itself has a very, very high resistance to the point that it's even difficult for water to get itself through, When I was sitting in your seats, we used to think that water just flowed right through lipid membranes. It doesn't. They're aquaporins. They're large molecules that allow water to flow through. So nothing, if water doesn't make it through, nothing much can get through without it. Yeah?
So is there no concentration gradient between the cytoplasm and the nucleus? Can ions flow through the pore?
Ions can flow through the pore. And in fact, things like that calcium. The question was, is there an ion gradient? And obviously, the pore is large enough to let ions through. The double bilayer doesn't allow things through, but these pores are enormous. So it's difficult to keep a gradient. Previous studies which said that there was a calcium gradient and calcium was elevated in the nucleus, as you might imagine, were based on fluorescence studies comparing cytoplasm, which in these figures I've been giving you might be about a micron thick in that living cell, and the nucleus is about 5 microns thick. So let's all agree that if you make something 5 times thicker with the same density of indicator in it, that you will find that it looks like it's 5 times brighter, okay? That was that information. It was just an incorrect assumption based on the thickness of the slice. So none of the monovalence or divalence tend to be able to keep a gradient. The exception is there are obviously a lot of binding proteins inside a cell. Okay, and there are a lot, something on the order of equivalent about millimolar or two of BAPTA or EGTA is sitting there and represented by the cytoskeleton and about, basically vitamin D and these other diffusible binding proteins. All right, let's switch gears and go to that little thing I left you with, which was the nucleolus, shown in this next slide. Here's a bigger picture. Again, euchromatin here, heterochromatin there, and then a couple structures, one here and one there, that are very heterogeneous. And, back in the light microscopic days, we didn't know what it was. It just was this little tiny dot inside the nucleus. Here's a blown-up version of a transmission electron micrograph. And there are a couple things that we need to address. One is, we're anatomists, so we go name things. So there are basically three areas of this. And let's see if we can do it. There's a dense fibrillar component. Can you see my arrow moving? Here's my arrow. Here's this dense part. Okay, that's the dense. The blackest staining stuff is a fibrillar component of this thing. Inside some of this dense stuff, then you can make out a fibrillar center, which seems to be a sort of a homogeneous. Every time I show a movie, it decides to turn my arrow off. It is rather more homogeneous. And then there's a granular component that you might be able to make out. Let's go look up at this side of the slide. That is back to that lower density, has white spaces and things like that. Whenever you see some things that you can peer through, then you think, great, this must mean something is active and going on there. What's active and going on there? That's shown in the singular set of micrographs, I think, that were ever produced on this particular thing. And so, come to know and love this because you're liable to see it at some later date. You can just make out that there seem to be, a bunch of caterpillars following each other around on this extended smear, but they're not quite uniform in shape, and there seems to be a filament connecting them. Now, if I go down and I look at that here in the higher power view, then this little filament, it's right at the tip of my arrow right now, here, is chromatin, all right? So this is something that I want to transcribe this thing and do something with it. And then what we have here is what you're seeing coming off as we move along, getting longer and longer as we go from left to right, is basically a polymer that's being produced. In this particular case, it's the nascent ribosomal RNA that's being read off from the 5' to the 3' end, all right? So that means that these little polymerases are moving along the chromatin from right to left, okay? Because as they go on, the molecule gets longer and longer. I'm sorry, let me try that again. From left to right. No, I was right the first time. Here it is just beginning, and it's short, and by the time we get down to this end, it's very long. Excuse me. It's still moving from left to right. It's going along in this direction, and it's finally at the 3' end, and the 5' end is sitting up here as this little dot that's there. There can be verbal typos, and that was one of them. All right, as I mentioned before, this whole little precursor is going to be used in constructing then the ribosomal element that's out there in the cytoplasm. But to get it out through that nuclear pore, it's going to break this into a couple of components. And that's shown in a figure that's somewhat difficult to make out from Kurzenbaum, a text that we used to use, which shows you that same profile, but it's showing you, to confuse the innocent and me, it shows you going in the opposite direction here. Here's the longest ones. Readout is in this direction. The direction of RNA synthesis is going from starting at this end and going in that direction. All right? So here is the cartoon showing you these things getting longer and longer as we start out here at the beginning and are moving our way along the chromatin fiber. And then what happens is that it gets broken up into two chunks. And the 18S chunk and the complicated 28, 5.8, and 5S chunk get transported out of the nuclear pore independently, then combined to form your ribosome. All right, so they then end out there, in the cytoplasm or attach themselves in the ER. Yes?
Sorry, just back with the slides. You said the granular component is where like all of this is happening. Can you tell me on like the big one where that is? Like where this, this area.
Okay.
It's back to the slide.
Okay, so it's not back to the introductory slide, so we don't have to watch the movie again. The question was, could I show you back a couple previous slides, then where this higher power view, I'll go back to the higher power view, this higher power view, where is it when we look at it on a lower power version here? So if you look in this lower part or version here, you can see some things that are very black, okay, and down here, very black as well, with a gray center. And then you can see parts that are just gray and don't have a dense fibril surround, okay? So that's why we zoom, give it higher excitation voltage and raise the gain on these things so we can see this. So this brings up a question. If we have to have chromatin laid out there so we can go along and read it off and construct something like that, ribosomal RNA, this there. What does it mean in terms of this whole nucleus and all these parts that are there? Here I said it was really condensed, and here it's sort of open, and it might be, reading what's going on there. And I think it's probably worthwhile remembering that if you take a normal eukaryote human cell, you've got about 3 meters of DNA in there. Okay? So then we look back at this nucleus, and I told you already this thing might be 5 microns across. So there has to be some way in which I take 3 meters and I put it into 5 microns, okay? Some rational way of organizing that, and there has to be a bunch of tricks involved. Counted as a given, and Mother Nature has a bunch of tricks, all right, of which we know very few at the moment. But one of the ones we do know is that there are chromosomes. And if we look at in prophase, then we find that we can basically lump that genetic information. It's not one big loop like in a prokaryote, our normal E. coli that we can use and insert things in. It basically has distinct elements. And as indicated over here on the left-hand side, we have a chromosome. It has this constriction and centromeric region in it. It has a short P region and a longer Q region. And there are 46 of these with XX as the last ones in females and 46 with XYs in males. And in the point of basically being able to take some cell that's in prophase and staining all those, back in the late 60s, early 70s, people started being able to do karyotyping and looking. And what were they looking for? They were looking at exceptions to these rules, 46 and 46, and then sometimes there were 47, and sometimes there were more than 47, and there were various other supernumerary chromosomes that were there. And these were associated with genetic problems, and that will bring you to the second semester as you do the nuclear parts of biochemistry, also known as genetics and molecular genetics. Now, from the 70s, where all these things were basically stained blue, so that we could look at the sizes and lay them out and decide whether or not they look like a normal component set, then if we fast forward into now, what we come up with is a bunch of now ways of basically coloring with different fluorescent probes each one of these different chromosomes, all right? And this is good, because then we can label the different chromosomes and basically keep track of what was coming from the paternal and the maternal source. Yeah, we are going to talk about sex. We have these too, and we want them together, and we want them to exchange products, okay? But the first things first, we want to know where is it that they hang out in the nucleus, all right? So if we have multiple colors, instead of everybody just the blue man group sitting there in the nucleus, we can actually take a look at it. And so what happens is if we go up here, we can lay them out as normal from biggest chromosome to the smallest one, which is the Y chromosome. Y. Yeah. For those of you who have adolescent male brothers, you know why. Why is he like that? Here we have it. So we go through this entire series of 23 pairs, those 46 chromosomes, and there they are. And here they are laid out for you in beautiful taxonomy with different flavors and colors, and then bang, we put them back into the nucleus. And if you look over here in the nucleus, you find that, well, there are no regions really where the colors seem to be all together. In fact, we take this fluorescence micrograph and we go back over here to the schematic of it. You can take chromosome 7 here, which shows you something that looks basically in red, going up to here and over, and its pair is basically across the nucleus from it. Well, that's all well and good. I mean, you can imagine then when we get to the points of mitosis and the rest of the things, you want everybody lined up on the metaphase plate, that's not so bad. It doesn't really matter, does it, whether the maternal and the paternal stuff is there. But it does really matter when you're trying to sort things out and produce those gametes, all right? It's one thing when we have a normal complement of DNA and a normal number of chromosomes and the diploid number, and then it's another when we have a haploid number. And we want exchanges. We want to have some advantage given to us in evolution in terms of being able to recombine things in our chromosomes to take advantage of some variation that, depending on what the world hostile as it is out there is like, we can survive, okay? So from a molecular standpoint, the problem is that to do that, we need to get all these different chromosomes back together. So they have to be compact, because we know in aggregate they're 3 meters long, and they have to be the relevant components that we can get together. Is that clear? Not so bad. But just to reinforce this first issue of, how big is it, is here we have that prophase chromosome that's here, and the scale bar in this case is 1 micron. And here is just a smear of blowing up the nucleus and letting the DNA come flowing out strand by strand, and this is now a 10 micron scale bar. And you can see, basically, this is a very, very efficient way of packing the genetic information. So let's think about some of these problems that we need to solve. And let's think about what this very dense heterochromatin must be like and how we packed it up and what this might be like in terms of how do we functionally read it if it's packed up. I mean, how do we make it so it's flexible so if there's a transcription factor that comes, gets imported, it can go and attach to some part of this chromatin molecule that's there. Well, to look at it, we can start, and we want to start and look at the hierarchy of the levels of formation of condensation, if you will. As I mentioned earlier, it's going to condense this 3 meters to something that's 1/10,000th that size. So it's pretty good. So if we take a look at it, then if this is our idealized DNA, double helix, and here's the major groove, and there's the minor groove, and we set it running, then the first layers that we know something about then really come out to be the only layers that we know something about, because we've actually done crystal structure for it. And the crystal structure took, in the nucleosomes case, only about 9 years. So here's a histone coming down, another one, and we're forming a loop around of DNA, almost 2 complete cycles around it. And then we have a neighbor, and we have a linker region that's there. Okay, and this whole complex, with the basically 8 subcomponents of histones and the DNA, is the nucleosome. And it can then wrap and compact itself in and form a thicker sort of strand. So we've gone from one thing, which is essentially something, the DNA helix around these nucleosomes that are here, and compacted it into this unit that you see on the 3rd row down. Okay, so we know about this because Rosalind Franklin and her colleagues basically crystallized it down in London and got scooped by some guys up there in Cambridge. And then the guys up there in Cambridge, later in the Laboratory of Molecular Biology, went and basically worked out this structure. That was legit. And then this structure is, as I'll show in a minute, just a plausibility argument of how you could pack a bunch of nucleosomes together. From there on, taking this whole thick fiber and throwing it into loops and then twisting it around and compacting, as it's showing it here. Here is the phenomenological endpoint, which is this thing that if we compare this length to this length gives you something that's basically this reduction by 10,000. Okay, so the compaction into chromosomes that are recognizable is a really highly significant thing. There are 2 main morphologies that we care about. Their images extant in the wild of electron micrographs, and those are shown In the next slide, this first one, the beads on a string, and the second one, the 30 nanometer chromatin fiber. Shown here, here are beads on a string down at the bottom. You can see each of one of these. That's that almost double loop of DNA around the histones. And then if I compact these all together, I end up with this sort of 30 nanometer particle, more or less, going along up here. Now, obviously, to analyze what the elements are before the actual crystallography was done, biochemistry was done, and there's basically you can digest away the total from the end of one nucleosome to the end of the next is about 200 nucleotide pairs. That's how much DNA there's there. You can digest this linker, and you come down to a number that's on the order of in the 140s, I think, of nucleotide pairs. So that's the number that wrap around this histone core of the nucleosome. It's a rather small particle. It's not much bigger than the distance across the lipid bilayer. But as I mentioned, after a great deal of work, then the crystal structure of the nucleosome was actually completed to various levels by two different groups. And what's shown here in color in the center then are the four different components, the four different histones that are seen, four on each side of this. And then in the gray, wrapping around it, is that strand of the double helix, which is how it basically organizes itself together. If I put the schematic together with the crystallography that's shown in this next one, and here basically shown with the histones with their tails sticking out, and in biochemistry, you'll go through the ways that you can modify the histones and what the impact is on transcription, et cetera. Here is, again, the crystal structure, though, seen from the side. So, as I mentioned, a lot of this is being done by the biochemistry of it, and we can basically separate out what is the core from what is the DNA. And here's the answer: it's 147 nucleotide pairs that are in that wrap, double wrap around the histones. And we can analyze how they differ in case of the histones and what their significance is. Now, one of the things that we're interested in is that whenever we do structure like this, in the back of your mind should be that function question, right? I mean, how does this thing work then if, okay, the DNA is bound around these histones, and then what? How do we get to it and talk to it? Well, I don't have, there is no movie of this, but there is the thought that one can do by substitution and asking kinetic studies, how long does this rat nucleosome exist? And it exists, as I've just described it to you, for about a quarter of a second, okay, about 250 milliseconds. And then it can throw a loop out and bring it back and rewrap, as shown here. And these are kinetic reactions that are going back and forth, right? And it can stay off for as long as 50 milliseconds, right? And it's during that interval that other things, the sequence-specific binding proteins, can go and latch on and attach, right? So this is the reason now, if you take and complete the picture of what we've been talking about this afternoon, you come in with transcription factors and the rest, and you import them specifically to go and to be able to look and to bind on to a particular sequence that's there in the genome, to turn this thing on and to get the results. And this is what is the whole response sequence between the environment for that cell and its response in terms of what it produces. As I mentioned, the part that we know anything about then, it can be represented on this particular slide of trying to put something together. This was the initial model that was there. After more work on, this was supposed to be the 30 nanometer fiber, if you will, by having these guys just fold as pairs and going through it. Obviously, without evidence before crystallography, that was good enough. With the crystallography, though, then this is a different suggestion. The red, white, and blue then give you positions along this particle, give you the folding, and over here on the right-hand side is the crystal structure of that 30 nanometer particle. So the take-home message is where I started out with this for you, and it's looking basically at how it is that you can compact within that nucleus the genetic information into some size that's reasonable. And reasonable means that it fits within a 5-micron package at most. Now, back a couple days ago, Dr. Brady gave you a tutorial on mitosis, and the rules basically that those cytoskeletal elements and the kinetic elements of molecular motors were involved in this idea that you had to have replication of the genetic material and then separation out into daughter cells. Okay? All well and good. In this particular case, then, if we look at it, this is a DIC image of a fibroblast that's sitting there, and you can see that it's coming up into prophase. We're starting to get some identification of the various chromosomes that are here. There's still a nuclear envelope that's around the outside. And if we let the video play, then this will go forward, and we'll see all these chromosomes line themselves up on the metaplate and then subsequently get divided. So we have an organization process that's going on here, and we can see basically that there is some area where that's central to it. Each of these are the 2 chromatids. Each one of those red arrows are 2 chromatids of the corresponding chromosomes. This is the metaphase plate, and unmarked to you was then the traction along the microtubules to bring you to the 2 daughter nuclei that are shown here and here. Then we're going to have a kinetic phase in which we're going to pinch off the cells into 2 cells, and simultaneously we're going to reconstruct that nuclear envelope. You know, it's like one of those things you think, boy, if I slow that down, can I actually follow that? And no, you actually can't. There are so many things going on simultaneously and so much signaling that there is still much to learn in my lifetime, to say nothing of yours. At this point, then, sex does raise its ugly head about what's going on here. This is a scanning electron micrograph of that infamous moment of conception that's been so popular in the press recently. Just to put numbers on what that means, the moment of conception, if we take it from the moment of fusion of the sperm with the egg wall and somebody actually making it in, to the moment when that zygote ultimately divides and we go from single to 2-cell stage, that takes about 23.5 hours, which really redefines the term moment for me, like a moment of conception. To me, it takes all day, and I know the initial act didn't take all day. I'm willing to entertain it, but my wife isn't. But so we have this problem in that it has a great deal of success. You know, we've only been out here screwing around, so to speak, for about 10,000 years and recording what we're doing about it. And so we've come a long way in these last 10,000 years, believe it or not. And part of it, significantly, is distinguishing between species that use sexual versus asexual reproduction because it allows for a recombination of the elements that are there. So I want to take a look not just at the nucleus and at the packing of chromosomes, but I want to look at what's going on here. And I think it'll give you some insight into what goes wrong and why you have various, you'll see them next term, various birth defects and the rest, okay? It is because it's a very complicated, very slow process that has to allow for exchange of information between what were those maternal and paternal components of the genome, okay? So sit back and relax because, you're going to see this in some other arena, but what I want to do is give you the structural basis for it. So first, over on the left-hand side here, we have basically the organization for a diploid organism versus a haploid organism. Both of these actually end up using meiosis. Now, meiosis is sort of like that stepchild that you don't talk about except on the step 1, okay? Because on the step 1, they're going to want to know, at this phase, how many chromosomes are there and how many times the normal amount of DNA, okay? It's guaranteed. It's guaranteed. And it isn't there, it'll be there on the shelf exam at the end of the year, okay? So it's just something you have to know. You will deal with mitosis a lot. You'll deal with meiosis now, all right? So come back to this later. So if I'm a normal diploid organism, that means all my somatic cells, which are highlighted in blue here, are, they have this basically 23 pairs of chromosomes that are sitting there. And the only thing that's different, oh gee, every time I run a movie I get to reset this, is the germline cells. And the germline cells for diploid organisms are haploid. It's, you know, you have half of it, and now you have to get together, my little half-wits have to get together with somebody else to create a whole zygote, okay? Well, that's okay. I'll take the burden. But the question is, how do we create a haploid cell, either the egg or the sperm? And we're going to go through what that looks like right at the end of the course, okay? When we're in ovary or when we're in the testes, we'll take a look at that. But the important thing is that the eternal cell line, your germplasm, these things that really do look like, you know, the only eternal cell lines you know about are germ cells or, you know, stem cells, really the derivatives from them, are basically unchanged except for these epigenetic things that I alluded to earlier. Right? You can change how your germline comes out by modifications of the genetic information, but the genetic information, on wood, if it works right, will be preserved and replicate what's there. So what we have is, for any diploid organism here, then, we have a germ cell line which undergoes meiosis and creates either the haploid egg or haploid sperm. During fertilization and that day of bliss, they come and create a functional diploid zygote again, and from that point forward, it just goes through the stages of mitosis and then produces yourself an organism. Fair enough? Straightforward. If we have a haploid organism, then, you know, basically we just change the timing on this. We take two haploid organisms, we mate, get a diploid zygote, which it doesn't know what to do with. It undergoes meiosis then and creates a bunch of haploid cells, great relief, and then undergoes mitosis with that haploid number and produces basically the subsequent organisms, all right? So as you might imagine, those of us who are up walking on hind legs talking, you know, tend to be diploid organisms, and those guys occupying our gut and doing things like that tend not to be, all right? So there is a figure, and this is just one of many, and you'll find it in every book you own, with the exception of Moll Flanders or such. And this is one that shows the cell cycle. And what it normally does on the cell cycle is it ends up here in mitosis. When it has two centrosomes that have been produced, you know, a cell starts out with one. It has to duplicate it has to duplicate the genetic information, and when it has double the amount of each of those, in other words, two centrosomes, then it can undergo this cell division and subsequent cytokinesis to produce two daughter cells. So the action for mitosis is right up here in the top right-hand corner on the northeast here, which shows you basically what we just saw in the differential interference contrast movie of, you know, cell division. Bog standard. What I'd like to do is contrast and compare then mitosis with meiosis in terms of what it is that you have to get your head around. And the first thing is homolog alignment that's there. Now, during mitosis, we have homolog alignment. In other words, if we have here indicating that this is replicated chromosome 1 from the paternal side in red and the maternal side in gray here, the centromere. So one of these in one color is basically their sister chromatids. The pair of chromosome 1 from mother and father together is known as a bivalent. All right? And it's just those two of them together. In mitosis, there is no crossing over. There's no confusion between the pair that was paternal and the pair that was maternal and whether or not we get an exchange there. There's no advantage. There is, the advantage of sex disappears in mitosis if everything goes correctly. All right? And in mitosis then, basically for the maternal and paternal pairs, then its center here, the two sister kinetochores from of the two chromatids then attach to two different centers and, as you saw before, get pulled apart. And then in one side, if we take it around this, it gets packaged in telophase into one of the daughter cells, and on the other side, this pair gets packaged into this one. All right, that's straightforward. But imagine what happens in meiosis, and then this is where basically all hell breaks loose, okay? because we go from this state, which is shown over here on the left-hand side, and then we have an exchange of genetic material between what was the paternal side here in red, grabs onto the maternal part gray down here at the bottom, and vice versa. Over on the maternal side, it grabs the corresponding lower piece of the genetic information that was initially paternal. Okay, and this crossover, as you'll see in neuroanatomy, is a chiasm. All right, you have an optic chiasm and all the rest of these things. Well, this is a genetic chiasm that's going there. So these chromatids are bound together very strongly with each other, and this pair and the bivalent are bound together as well. Strong enough that basically what was a big polymer on one side of the divide is eventually going to basically be able to cross over. And it can do it not just in a single place as indicated schematically, but in here, in using not human, but using a grasshopper chromosome, you can see that there are 1, 2, and 3 places where we have crossovers taking place. And so the paternal here in red and pink is shown there, and the crossing over gray to the maternal then shows you where a molecule has basically taken on the opposite parent's genetic material. All right? Now, there's, as you might imagine, if it took a long time for information, the genes to get from various parts of the nucleus to be together to have this conversation, it takes even longer for the splicing and all the rest of it to get done. Okay? And that's one of the things that's going on as you're producing, in our case, these haploid nitwits that get used to actually create a zygote. All right? There is a long intermediate period, which can occupy years, of where this crossing over can take place. The other thing that you should be aware of, obviously, if you have something with this sort of complexity that's going on, there can be mistakes. All right? This is relying on molecular machines essentially to cleave stuff, allow it to be reannealed together, and to go forward and get reproduced. Okay? So there's a lot of steps in the process that can go wrong. Before we actually deal with those things going wrong, though, I just want to give you a schematic of what it means and what molecular elements we know are there. Over at the top here, then, is the filaments that are there of basically one of the homologs, and here's a second one. And what we've had to do molecularly is not only tie together the two corresponding ones for either the father or the mother, but we've also had to tie the two together so that they can have this conversation. Now, what does this look like in terms of what you see when you look down the microscope? And in this next slide, which is fairly complex, I'm just going to compress all of the time, that from the left-hand side to the right-hand side goes through all the stages of this sort of a mixing process. And this is showing this process of bringing things together, that you're bringing the two homologs together, is called synapsis. In the same way that you have synapses between cells, that you have a connection, a communication there, this is a synapse between molecules that's there. And what happens is we start out with 4 chromatids, the paternal and maternal ones here, and first we bring them together and make them so that they're locked together, and then ultimately we bring the 2 pairs together. And that's when we allow for the exchange of information from a paternal segment to maternal and vice versa. And then at the other end of the process, as time goes on, we obviously have to separate these off into 4 separate chromatids again. Fair enough? All right. So can we see this process? Yeah, we can see this process. And here we look at, basically, we have chromosomes that have regions that have not synapsed, which are shown here by the red arrowheads. And this is a process that goes on, it's one thing to use grasshoppers, it's another to use fungus, right? But it goes on across all these eukaryotes, so it's worthwhile. So in this case, we have it, they're partially synapsed, that they haven't joined together completely in these chromosomes. By the time we get out over what's in panel B, they're completely synapsed together. And so at that moment is when we can have the exchange. So do we do this in humans, or is it just fungus and grasshoppers? And the answer is yes, because there are crossovers between the homologs, and this is using testes. And in the testes, then, if we look at each one of these red areas, these are the synaptolemal complexes, the synapse being brought together. The centromeres are these things that are in blue in each case for it. And what are regions that are green are areas where it's been crossed over with a corresponding part in what was originally, for this male, the maternal component of his genome. So to actually come up then, just two more slides. First, this is the one of mitosis, which you've all come to know and love, where we have this center, which basically we have an attachment on the two chromatids from a single parent. We're attaching to the centromere, and we're pulling them in opposite directions, both for male and for female here. And then we end up with these daughter pairs. Now that process is very similar to, from this point in this diagram, over to the right. Okay? Because in this case, we have these two chromatids that are attached to two different centers by their kinetochores and pulled apart. They're going to be pulled apart so that they have haploid daughter cells. So meiosis can be thought about as two processes. One, an initial one that allows for crossing over. Okay? And the chromatids, parental chromatids, are going to both, in the male's case, be attached to the same center and go in one direction, and the female, same center and go in one direction. That brings you to this point in the middle of the diagram. So metaphase 1, basically the things that look like the maternal, genetic information, both go off toward the bottom of the screen here. The paternal information goes off toward the top of the screen. What is critical then is at this moment, right here, we still have all this crossing over between paternal and maternal information, all right? And it's at the moment of this being translocated that there's basically enzymatic breaking of all those cross bridges, all right? So that's a point at which, if everything goes perfectly, these separate off into basically lumps of genetic material that are the right size and appropriate for what they're doing. Then at this point, we have a pair of chromatids, though, and the only thing that remains to do is to have them attached to a kinetic center again, and this time get pulled into opposite places and put a wrapper around them. All right? So we have two different ways of cell division here. One is universal, and you'll see during the course of your patients' lives all the time. Sometimes it goes out of control, and we call that cancer. Sometimes it's kept well regulated, and everything's fine. The other form of cell division is meiosis here, and it's very important if you consider what can go wrong. All right? And you consider that the cellular events that are going on have a significant rate of error that, if we were able to track the rates of spontaneous abortions and things because what's developing isn't actually genetically correct, is a very high rate. When I was in your seats, it was thought to be 1%. Now it's thought to be 40 to 50% of failures of implantation, and the rest are because the embryo, as it develops, isn't perfect. And why isn't it perfect? It starts back out at this phase, usually, in terms of meiosis and the very complicated mechanisms of genetic sorting that are taking place there. And with that, I will give you a break, and you can come back at 3 o'clock.
Hello? Carefully disguised as an on-off switch. All right, and it ends at 4:20? I turn into a pumpkin at 4:20? About 7 hours and 40 minutes too early? All right, I guess we can continue talking here. Well, let's talk. How are things going? Have any questions about what's going on so far? You're getting a lot of stuff thrown at you. You notice that? Fortunately, it's not difficult. There's just a lot of it. Just a lot of it. It's all about coping and time management, folks. You'll get used to it. You'll get used to it. I want to emphasize that you must have ROS version 6 edition. Unless you want to be more confused than you are now. Because when you go to the manual, all the page references and figure numbers are to the version 6. And version 6 is distinctly different than version 5. It's quite a bit better, in fact. One thing that I think Dr. Art mentioned earlier is that we may be the only histology course in the country that is so integrated between what we have access to as a resource in terms of physical glass slides and now virtual microscopic web slides and the textbook. Turns out that the principal author, Mike Ross, who is now departed, was once given a class set that we used to hand out by a former department head. That's okay, you know. I know Mike Ross; he's a good guy. What we didn't know until a few years later is that he basically took photographs of a lot of things in the slide and put them in his textbook. So this became obvious when I looked at some of these images, and man, you know, that just looks just like one of our slides. And sure enough, I wasn't confronting him. In a bar one time, he says, "You know what, Mike? You know, you really did take a lot of pictures and put them in your book from us, didn't you?" Yeah. I says, "You did." So that textbook has a lot of the images in there are from the class set, and from that class set, in a large part, we've derived the digital web slides. All right, there's a question over here that I ignored.
I was going to ask, how does it work? Because so I know that obviously there's different lectures and everything like that, but like, how do you individually contribute to like the testing that we do?
We write questions.
So you just like write like up questions for the sections you like, or because they also talked about how it's supposed to be integrated between campuses and things like that.
Well, that is a more higher order question you have to ask from Dr. Art. But yes and no. Locally, we of course write questions based on what we teach you, and based on what we didn't get around to, but still in the textbook, and what's in the lab manual. To the extent that is not shared between us and the folks down in Urbana, it still holds. And the content we teach here, you have to ask more about that, how great that's being integrated for the first exam. Do you want to comment on that, John? How much are we integrated with what's going on in Urbana right now? Is it November yet? Is it November yet?